or his colleagues can contribute some compounds of this project, which was about Chagas or Leishmaniasis drug discovery, so some sort of that.
I don't have that on the. So did we speak about that last time?
I think Kimberly was interested in such compounds.
Uh-huh.
Yeah, it rings a bell to me as well, because it's one of the lead series we have on the GitHub site, of which we are interested in. And I do recall that we would have reached out, I guess, to see who has more of these molecules.
Right. I forgive my memory, but I couldn't remember if it was the benzimidazoles or the aminothiazoles, series 5 or series 2. I recall the conversation now, but I can't remember which project it was for.
The benzimidazoles was the ones where we would look into if there's other molecules, because I think fenbendazole, etc. There's a close link to one of the other projects from DNDi.
Right.
And the aminothiazoles they were making with the students.
Yeah. So the idea was to reach out to Ben and DNDi team about benzimidazoles.
Yes, he has a project about benzoxazoles. So I was not sure if they might be also active against this mycetoma fungi.
Right.
Can be a chance.
And Wendy, as far as you know, of course, we've evaluated some based on the boxes that have been evaluated, but we haven't done a targeted screen for benzimidazoles, right? Following that?
No, but those were the ones which were most potent, because out of the 4 or 5 tested, 3 did prolong larval survival.
Yeah.
And I think they were also active against Falciformispora senegalensis, and that was the only category which was active against both pathogens we screened.
Right. That's right. No, it's looking very promising, I guess. Okay, so is that action on me or Wendy or somebody like that?
Of course, I can also write him an email if you want to.
Okay, maybe CC me and Wendy in, just to make sure we're all looped in. Great, so we can put that as an action on you, Bernard. Great.
Okay. Yes.
Thanks for reminding us.
I see that Hang is here, so I guess Hang, you're taking the minutes.
Yes, I'm taking the minute now. So Kimberly and Alice will be an apology for today. They just had a very big breaking good day with with the students, with the SSP students, that they just want to take a rest for tonight.
Okay. Yeah.
Great, thanks. So just to, we were just talking about Bernard is going to send an email to Ben Perry to ask about availability of samples of benzimidazoles and related compounds to see if we can get some things tested. Okay, great. All right, well, I'm going to work my way through the agenda, which is issue number 76. Just put this up here in case people don't have that. So that's the link posted in the chat window in case you wanted to follow along here. And we can whip through these things. Some will take more time than others. So the first thing really was about the paper writing, and just to make sure that we don't have any kind of bottlenecks, right? So we know where the next action points are. And the files are shared, and the current version of the paper is in there. I guess does anybody have any comments about the current version of the paper and where we're up to? I had a feeling that Kimberly had made a bunch of edits, but has anyone looked at the paper in the last few days? And
I do. So I just want to give an update as for, so for my section, I did an update on the SAR. So I drafted the, I got a review back from Peter and Alice, like the last time, and then I made some edit, and then Kimberly made a review, and I just made an edit based on that review. So the latest draft of the SAR is now up onto the file. So I updated the file with the latest SAR information. So that's where I'm up to. So yeah, that edit should already have myself and also Kimberly's work in there for this.
Great. All right. So based on that, Hung, what do you think is the next step? I mean, the next section that needs to be done. Do you want that checked? Or you know, what's the most, I guess, unready bit of the paper now? You're muted.
I would like to have someone else just have a read through that as well. So just double check that it looks okay. That's still a bit on me. I've left a few bits for supporting information. So once we kind of just agree on like what goes in and what goes out, I'll finalize that. So right now it's just a comment that some of these will not be in the paper, but I actually haven't created a document of supporting information for that. But
no, that's fine.
If someone can check it and make sure like we're all happy with it, then I'll finalize that as well.
Okay, great. So it's a free for all. Anyone else can go in and check now.
Yeah. I don't know if maybe Matt or someone has a preference for checking, but otherwise, probably someone who's probably more like has experience in SAR analysis. Or more.
All right.
Anyway.
All right. And if anyone does make edits, of course, this is a Dropbox folder, right? So if anyone makes edits and changes, then save as a new version, right?
Yeah.
Mm-hmm. Okay. And how is the you know, I guess the chemistry characterization side going, right? So you had a meeting to decide on a consistent style, and you came down on a basic set of you know, agreed things on how we're going to do the experimental write-up. How's that going? Do you think the data is going to be in a consistent style? Yeah.
Okay. Yes, I am still working on that. I was planning to do that last week, but I did have an emergency ethics application I have to put in, and I haven't got a chance to do that yet. But I'll finish that tomorrow afternoon. I'm saving some. Setting aside some time to just finishing that up. I'm still working on it. But the thing that yeah, I took note of what we discussed. So we're gonna make that consistent. But I do have a point regarding the fluorine NMR. Some of my compounds actually don't have like the fluorine NMR. So I think if Dimitri hasn't done his one, I would suggest we just leave that now. I don't think I'll have it in time, like in terms of like have a timely update on that. So if we can just leave that out, I can re-characterize my carbon to show the splitting of the carbon fluorine coupling, which does in that case it's already showed that there's a fluorine there. So we can probably skip that if it's you know, if
yeah, we agreed last time that we would skip this because, for example, from UCL side, we haven't done any fluorine NMR because it was decided it was like not very useful at the time.
Okay, so I'm happy to just leave the fluorine out and just do the fluorine carbon coupling, recategorize.
Yeah, that's what I've been doing as well. So like from my side, it's like around like 60% like complete, like CF coupling for the carbon-13.
Yeah.
Yeah, we definitely need that coupling for sure. The thing about the fluorine NMR, I think, is not terribly useful if all the compounds are very similar. I mean, that's the basic feature here, is that the fluorine is on a ring that is almost always the same. But certainly in the carbon NMR spectrum, which we'd expect to have for most compounds, yeah, we need to be clear about the coupling.
Yeah, so I'm just gonna do that. And also in terms of the LCMS, and I haven't actually run an LCMS or any of my 17 compounds. So it might take a bit too long because I don't think I'm gonna have the time resource to do that. So I think Matt, you suggested that we can also submit in these NMR spectra themselves as a substitute for that. Is that okay to still do that?
Yeah, so at some point you need to prove purity.
Yeah,
that's the crucial thing here. And that is, there are multiple ways of doing that. In old school would be that you would submit elemental analysis, right? But few places do that. The alternative, which is very medchem, is to submit the LCMS trace, which is also good. And if we don't have that, then the other way of doing it, which I think we'd need to, would be for in the supporting information, we give the 1H and 13C NMR spectrum reproduced as evidence of purity, right? So normally you would reserve that just for novel compounds. And they're certainly needed for novel compounds, right? So for a novel compound, you'd expect proton and carbon NMR reproduction in the SI. For compounds that we want to prove purity on, that we don't have any other sense of purity on, then the NMR spectra will do that. You can't really put a percentage value on it from an NMR spectrum, but it does the job to demonstrate that the compound is basically pure, because of course the baseline is flat and there are no other peaks there. So that'll do. So if you haven't got the LCMS traces for those compounds, I would suggest just putting the NMR spectra in the SI.
Yeah. So
for novel compounds, I mean, certainly put them in anyway, right? So for anything that's novel, we'd expect them in there anyway. The thing you mentioned about the files, the raw data files, that's just a feature of the fact that we're doing an open science project and we are, electronic lab notebook users, right? So for any novel compounds, we're going to need the proton NMR and the carbon NMR data uploaded, as I mentioned on the GitHub issue for this meeting, right? That's important because it's a scan of the spectrum is fine for a human, right? But we're better than that now.
Yeah. So I can organize that because I have all of the files in the spectra anyway. So I can do that. I just don't think I have enough time to do all of the LCMS. It might take a bit too long. I would prefer to work on the series 5 for now, which is what I'm focused on, because I still have the series 5 and also the other project for chemistry education research as well. I just don't have enough time to do all of it like in one go. So
Right.
yeah, I'm happy to prepare the files for those one as substitute for the proof of purity and also for the novel compounds as well.
Great.
What if we have a 50-50 split situation? So one side has LCMS, because at UCL we have LCMS machine, so we can do the purity as initially we were planning to, and on the other side Han doesn't have time to do the LCMS. So do we scrap the whole LC thing for UCL?
No, we would include that. It doesn't matter if it's not completely consistent, right? Different places are characterizing.
So it will be incomplete set of information for the LCMS. Is that correct?
So some compounds will have LCMS and some wouldn't.
Yeah, and I'll double check with Kim whether she wants to do LCMS for her compound, because she mentioned that she would be interested in doing that. I'll talk to her and see what she wants as well.
Okay.
Great. Okay, great. All right, next item would be potency data. So I think for Ma, there was an item here on you to check and update the in vitro potency list with correct compound classes, mainly series 6 photoxemes. So to check which compounds were belonging to which series, because I think there was a slight confusion there last time. Ma, did you have a chance to look at that?
Yeah, I think all issues from my side, I can show you all the slides. It will be more easily to talk and share. Would you mind sharing my screen with you? Or?
Sure. So hang on a second. Can you? Yeah, there you go. Good.
Can you see my screen?
It's coming.
Oh, yeah. Slides.
Yeah.
Okay, so the screening compounds from Mycetoma Open Source 2022. Yeah, firstly, the compounds from our side, natural products and series 1 compounds, and the 5 compounds from series 6, and 8 compounds from Kenya. As for natural products, finally we get 4 compounds inhibit the fungi growth. And we finish the IC50 min. Because these two compounds are liquid, so we use the volume dilute volume. So then we did the toxic test in larvae, and the petrohemon is toxic at 20 micromolar. So next step, we want to use the 10 micromolar to test whether it is toxic. And this one, nafesarin, is not toxic. So next step, we will perform it in larvae. And this is compounds from Mycetoma Open Source, including series 1 and series 6. The MIC for MM55 is well finished. Yeah, and the IC, we selected the IC50 below 8 micromolar. Then we decided to do the toxic test first, and this one are not toxic. The further test will be performed in larvae, and the others, yeah, we'll do the same test. And these three compounds from Dimitri side, I think, and I already updated the those data have been updated in the spreadsheet yesterday. Yeah, so you all can find the data. And this is compounds from Kenya. I received 8 compounds from Kenya, with the note as PTM, PTW, CDM, CDW, like this. And these two, fetal taxponent and SA, this two is volume. This two is liquid. These two are the liquid. So we use volume, dilute volume, to do different concentration test. Then none of them inhibited the fungi growth. Yeah. In the near future, in vitro test, we still need to finish the FIC 50. Yeah, for those selected. We selected with the IC50 below 8 micromolar. Yeah, and the vivo test, we will finish. We want to finish the therapeutic efficacy test from the last season. The two phenobarbital analogs, MycetoS 166 and MycetoS 174, and one ketosme is the MycetoS 193. And this season we select totally 15 compounds, including 4 natural products, 5 ketamics, and 6 phenobarbials will be performed in larvae. So yeah, that's all.
Right. Just very roughly the timeline of those near future in vivo experiments, very, very roughly.
Sorry?
How long? So the timeline for the in vivo experiments, very, very roughly. How long do you think those in vivo experiments will take?
Yeah, this week I will start to do the first time infection. This week, maybe tomorrow. Yeah, because larvae will come tomorrow. Then
okay.
Yeah, and I prepared the new flasks, the new fungi in the flask, and I think it will be finished soon.
Okay. So do you think maybe by our next monthly meeting we may have data?
Not from the 3 replicates, because we do these experiments in threefold.
Yeah.
But preliminary data hopefully we will have, but it depends a little bit on how the experiment goes, because it's the first time we infect again with Madurella mycetomatis this week.
Right.
So if all the controls go okay, then it can go rapidly. If the controls are not okay, then we need to play around a little bit.
It really depends on the experiments goes. Yeah.
Okay, because on the screen there are 8 phenaramols, right? Which are being evaluated in vivo. And I'm not sure if those are currently in our draft paper. Are those molecules in there?
No, I didn't think so. They're like because they came with the latest batch. They have not been included.
But they are phenaramols, right?
So DM41 to DM48 are phenaramols, and everything else. Well, actually, it's a mix. Sorry, it's a mix. It's just like series 1 are phenaramols and series 6 are catexymes.
So on just go back to slide 7, Mark.
Mark, could you go to slide 7?
7. Okay.
So the MyOS 166 and 174.
Yeah, those were left from last year.
Those
yeah. From last season, we didn't finish.
Right. Are those in our current draft manuscript?
I think they are.
I didn't put them into. Oh well, I had in vitro SAR on them, but because the in vivo wasn't there yet, so I didn't include them in my SAR analysis for in vivo. But I can add that in, and I think we should, because it's just like one of the hole in the SAR that I would like to fill that we didn't have a chance to do it last year.
Right. I mean, it's just if we have data coming, then that in vivo data coming for compounds in the paper, then it's going to help for us to wait for the data. Right. I mean, it will, whatever it is, it's going to add to the current manuscript. Right. Yeah.
But otherwise,
so
otherwise, I think probably okay to leave it out. But if we are still working on it and that comes in, then that would be great as well. But yeah.
Yeah, I agree.
I just quickly checked on the Mycetoma spreadsheet online. So those 2 phenaramols are Hanks compounds.
Yeah.
Okay, great. Very exciting. Thank you.
But I think it's a good idea that this larvae season will be the last to test phenaramols, because otherwise we will be stuck with only a few data points next year. And I don't think it's wise to prepare another phenaramol manuscript with only a few compounds.
Right.
There are a lot of ketoxamides also active, so that's maybe, well, the part Dimitri likes to focus on, and maybe if other compound series have hits as well, we focus more on those in the upcoming years than on the phenaramols.
Agreed.
Yeah, so from UCED as well, so we're probably working with the undergrad students more on the aminothiazoles, and myself will be working on the benzimidazoles as well. So we're happy to see if any of those turn up any more hit, and we can work from there. But yeah, I will be like pausing on the phenaramols as well. So that's all good.
The only residual thing is, so when we've completed the SAR analysis for the paper, and we have identified, the most promising compounds, and we've identified any holes in the SAR. The only thing we have to be careful of is whether there are compounds in the original EpiChem library which it would make sense to evaluate, or which would fill those SAR holes. Now, I searching in a fuzzy way like that, the EpiChem library is a little difficult, but if you remember back to the original stuff we got from that library, we only requested, 25 plus 25 compounds or something? I think it was 2 rounds, and there were, I forget how many 100 compounds there are in there, 600 or something?
So 800.
800. So there's a risk that, because we haven't looked at it for a while, there may be compounds in there that make sense to have a look at. So I think we are going to have to go back and have a little bit of creative interrogation of that database again?
I did have, so I had a look at that already after like the very first time I did my SAR, and I did pull out of, what was it, 6 or 7. I put it up onto GitHub, but then I got distracted and I actually didn't send an email to Wendy to ask for those, so I will. It's on GitHub in one of the meeting, like underneath, like on the meeting GitHub issue. But so I've done some of that already. But if anyone would like to have another go at that, I'll be happy to as well. Just make sure that it's not doubling up on what I've already shared in GitHub.
All right, that's great. So if you can reflect this in the minutes and then just point to where you've done that analysis already.
Yeah.
I think it is something we're going to need to check a little bit.
Yeah, and we still have a little bit of time if it's only a few compounds.
Mm-hmm.
So, but I don't think Ben would be very happy if we ask for 6 compounds now and in 2 weeks' time for another 6 compounds. So we need to make it in one effort, and preferably before next month, to ask the request from Epicam, so that we can screen it very quickly and roughly for the IC50. And if the IC50 is below 8, we will include it in the larvae immediately, just to hope that we can make it this larvae season.
Yeah, I mean, I think it's the easiest way for the chemists here to do that is to download the whole thing and copy and paste all the SMILE strings into Data Warrior, right? And then start searching around for similarity to structures that we think are interesting and see what comes up.
Yeah, so what I did when I looked through the library is I just look at the actual structures of those and just see what was interesting, but I actually haven't looked at like the other parameters. So I was just looking purely at like electronics, steric bolts and structure in general. But if someone else would like to do more like log these or, you know, other parameters, that I think that would be quite good to complement that, so we'll see if anything else comes up.
Yeah, similarity searching to best compounds and similarity searching to holes. That's all we need, because you don't want to publish a paper and then have, you know, the Epicam guys come and say, well, you know, that compound you're interested in, we happen to have that. You know, why didn't you ask us for it? That would be embarrassing. All right, great. If that can be put as an action for someone on the minutes, we can decide who's going to do that search. All right, thank you, Mar. So just one quick question, Mar, the data that you presented just then, so for the isolated compounds and for the extracts, are the data all uploaded onto the master list?
Yeah.
Okay.
Yeah.
Great.
I did that yesterday.
Okay, thank you. Perfect. How are we dealing with the extracts? Are we just given them MILES codes? So the Kenya extracts are just sort of natural product bits and pieces, right? We don't actually know what the molecules are. Is that right?
Yeah, they are mixtures.
Right.
Yeah, that's also for us a little bit, I don't know where to put them. We could make a separate spreadsheet of natural compounds instead of including them in the pure compounds. We could also just upload them in the pure compounds, but I'm not sure what's handy for the chemistry purpose. For us, it's just a compound or a mixture, but they will have, that's the difficulty. They will have, of course, different concentrations, because the compounds are in micromolar and these are in microgram per mil or in percentage volume to volume.
Yeah.
So we need to highlight that anyway in the spreadsheet, because we cannot just simply put them under 100 micromolar, because now we know, but maybe somebody else will not know, and in the future that might be lost.
Yeah, I don't know what the best thing to do there is when we're dealing with extracts. My natural inclination is to avoid spreadsheet multiplication and just put them in as entries in the master list with codes. And then separately in a column to the right of everything else to have the data that were generated on them, and to have a note there saying these are extracts from so-and-so group, maybe with a link to the relevant issue where they were discussed. Just because otherwise we start getting multiplication of sheets and it can get complicated. It's not what the MyOS codes are designed for, but there's no problem in assigning a code to them, because there are an infinite number of codes that we can use. So I think unless anyone's got an objection, we could just enter them as entries in the master list, and not give them smile strings and so on, and have the data in different columns from the ones that we're currently using.
Yeah, maybe also where you normally would put the smile strings, there you can already indicate it's a mixture of compounds.
Yeah. Mm-hmm. Something like that. But it would, yeah, if we can just make sure that they're in there, that would be really great. Thank you, Mon.
Where do we find the original information so that someone can put those entries into the master list? Sorry. Where is the original information?
I think IPA has that, so I will ask IPA.
Okay.
All right. And then some mechanism of action stuff. So Dimitri is working on updating the wiki pages with what we know of. As far as this discussion goes, the main relevance is the phenoramol mechanism of action, and what's been done on the sort of parallel campaigns. We previously in these meetings spoke about the possibility of, because we don't have a structure of the target protein, but there are structures of related proteins. And there are structures in the literature, presumably with phenoramols bound to something or other. Although actually, no, I'm sure. I think actually there aren't, right? There are no crystal structures of the proteins with phenoramols bound, right? I think Hong, you figured that out.
It's all azoles.
It's all azoles, right?
Yeah, it's all azoles.
Right. So one possibility we were talking about was trying to take a homology model or a homologous protein, and trying to figure out by a simple bit of minimization and docking how the phenoramols might be interacting with the target. And there are lots of clues in the literature about this, so we can be guided by that. But we could build a little bit of an in silico model to try and figure that out. And so there are some people on the call from Bouea in Cameroon, from a computational chemistry group there, who are interested in helping us out. This is Fidela and T. Kang, who's at the University of Bouea in Cameroon. And I think there was some interest there in trying to do some of this modeling with some typical phenoramols and trying to figure out what was going on. Have I got that right? That you guys are interested in maybe trying to figure out what the molecules are doing?
Yeah, Matt, thanks very much for inviting us. We are very excited to be part of the project. Actually, beside me is Conrad, and we are sitting in the same room, so we could not connect several computers, otherwise there'd be a lot of echoes. And so Conrad is a postdoc in my team, and he's working on a number of projects, and one of them will be building homology models for these proteins, these target proteins, and then eventually conduct some molecular docking so as to try to bring out some clues about the structure-activity relationships. And also by my side is Raisa, is a master's student as well, and she completed her master's, so she's trying to have an internship in our group, and we hope that within the time we are going to work, we are going to expect to have some interesting results.
That's great. Welcome on board. If I can, while we're on the call, introduce you to Wendy van der Sande, who is the expert on mycetoma, and who will probably be your most educated point of contact for anything to do with target proteins. And if you you know, I know you've joined the GitHub site, so you've got usernames now, which is great. If we collaborate there and talk about, you know, how we're going to get the structures of the most relevant proteins and what we're going to do, then Wendy is most likely to be the person who can help you the most on whether or not the target protein makes sense and whether we're doing things right. Others on the call can help you with the medchem and the outputs of the docking, but in terms of making sure that we're, you know, asking the right question, I just wanted to make sure you were in touch with Wendy.
Okay.
Okay, and I do know at least which proteins are the CYP51, so which gene it is. So probably you need that information as well when you start building. We did ask for the crystal structure with Seattle, but I still need to send a reminder because it has been quiet for more than a month. But it would always be good to model and do the crystal structure both, so that we know how accurate everything goes.
Very much so, yeah, that would be great. The Seattle unit were going through a rather nerve-wracking period of getting funding for their next few years of survival, and so I think things have been there's been some upheaval there. So I think that's one of the reasons why they may have been a bit slow to get back to you.
Okay.
All right, wonderful. Welcome aboard. Thanks very much, and we'll correspond about, you know, targets and what we can do online. And yeah, Wendy, if you're able to give information like that, that'd be really great, so we can get going. There was just on the still on this mechanism of action stuff, there was a question about whether we can use the AlphaFold structure as an approximation of what the protein looks like in order to make sense of the data. I don't know the answer to that. The structure is, I think, as far as I'm aware, the structure of this protein has been predicted by AlphaFold 2. Is there a reason why we should be careful of that, or can we just try and use it?
Well, the enzyme CYP51 has an iron center, so does the AlphaFold take that into account?
Yeah, I mean, I'm not sure. I haven't looked at it, but I guess I wondered if there's a reason, a sort of a priori reason why we shouldn't use it. Like there's something about the protein which means that the structure will be inaccurate, but I don't know what that might be.
Well, it has heme.
Yeah, and so I guess it's there if people want to look for it and check out whether it's reasonable. You know, there's discussions around, you know, how accurate the structure is, if it has, yeah, a significant cofactor maybe which isn't there, or, you know, the enzyme is meant to be binding something else which isn't there, and that could some other substrate perhaps which.
Well, they did model for Aspergillus, and I think they recently obtained a crystal structure, so we could check if that's the case. I'm not 100% sure, but if that's the case, how good the original model was compared to the crystal structure, and that will give us clues, and we can just simply mimic what they have done. Because if the crystal structure they in the end found was similar to their prediction, then there's no reason to think that we can't do it. Because Aspergillus and Madurella are both fungi, so probably facing the same problems.
Yeah, that's very good, yeah. Okay, and then, okay, I think that was everything on mechanism of action, which is, unless I've missed something out. Wendy, last time, so item number 4 is about kinases. Wendy, last time you mentioned you submitted an application for funds to buy the kinase inhibitor library. Couldn't remember what the funding agency was you went for.
The Fungal Infection Trust. And they will let me know somewhere next week, I guess, as they said the beginning of June. I did apply for the Erasmus Trust Foundation as well for an extra library, but that's only a pre-proposal. So it will take some time if I know I'm through to the full proposal, but it will not be a lot of money. It's just to purchase a few libraries and to be able to continue screening, because my funding will run out quite soon for this particular type of work. So I need to find some sources where I can actually obtain some money for the larvae to keep screening.
Right. Yeah. hence the need for the paper.
Yes.
Okay. All right. Good luck with those applications. All right. And I guess there was the item number 5 on the agenda other series was that Kimberly was doing something with a high school workshop, which was today. So I think I saw some pictures on Twitter about this, where students were doing the final synthetic step of some syntheses of the immunothiazoles in series 2, which is great. And I think that the thing that we wanted to make sure is that we knew what the targets were, well, things that have been made, but also maybe what the targets were, so that Andy at Sevenoaks, if you guys are going to go for some of these molecules, which are, I think, a little bit simpler to make than the phenaramols, that there's no unintentional overlap. And that Kimberly is I guess uploading or revealing, you know, which molecules are under consideration.
Yeah, I had a look at the list. She's put a list on there, and it's different to the one we've gone for. Obviously, the one with us is because we're cyclical and we only see our guys once a week, and then we've had exams and everything. It's felt a bit slow. So we've only actually been able to make one this year, but we're hoping next year we can probably get this going a bit quicker. So I probably need to have another conversation with Kimberly on GitHub and kind of just tie out to make sure we're not crossing over. But these were much simpler to make, it felt like. Obviously, we don't know for definite if it's actually worked, but it felt much quicker. And I think we've streamlined the process a lot more now as well, which I hopefully mean with because obviously see them, we managed to kind of get it going a bit quicker with the groups. So just hoping, yeah, but it felt much easier to make once we got the compounds. So feels like it's a worthwhile process for kind of the high school level that we're working at. So it seems to be good, and we seem to be making different things. So that's good as well.
All right, fantastic.
And Kim also, I just checked my messages. So Kim just told me to tell everyone that today went really well. So we're really happy that it was a success. So we do have another session this Thursday, which I will join. So I'll join one of the sessions to help the students. We have another session, synthetic session next Tuesday. And then on Thursday, we're actually so we're going to be working behind the scenes to get all of the characterization data ready, like the raw data, and then we'll bring it to their school next Thursday to do a spectroscopy workshop with them as well. So that's kind of the plan. So it's quite interesting. Hopefully everything will go well, and we'll have another batch of immunothiazoles for when need to test as well.
That's awesome. Were there 200 students, did you say, for this thing?
Oh, I think it was 100. It's about 100. Well, she told me these spectroscopy workshops going to be 103. So I guess that's about how many students are going. I'm not sure what the actual number for the synthetic workshops, because I haven't been to one yet.
Okay. Just while we're talking about that, if you could forward a useful protocol for how you are purifying the molecules at the end, that would help us with the sample that Andy's going to send us, that I think we're going to have a go at putting through a biotage or something and doing chromatography on. But if you or Kim have an updated protocol or something that seems to work quite well to purify the final molecules, that'd be pretty handy.
For the immunothiazoles?
Yeah.
I can ask Kim about that.
Thanks.
How many compounds are there? From Sevenoaks?
Oh, from Sevenoaks. It's one, yeah. Yeah, it's coming in.
Oh, it's been dispatched already?
No, I think it's about to be.
Yeah, I'm going to send it off once you've done this, yeah.
Okay, we'll
grab it,
keep you updated on this, yeah.
All right, and then last thing was the updates to the perfume compounds. Have we? I forget where we're up to on that. Apologies.
Yes, fortunately, it's not arrived yet, the package from Givaudan, but I have already some more eugenol derivatives, which I bought from common chemical providers like TCI or Acros, Aldrich. These are about 7-8 compounds of the eugenol series, and cinnamon nitride as another derivative of cinnamaldehyde. That's what I have at the moment. I expect to get the Givaudan samples every day, but until now it hasn't arrived yet. Sorry.
Okay, no worries. Thank you. So I guess we're waiting on delivery of compounds. Are those coming to you or directly to Wendy?
Yes, they will deliver it to me, and I will prepare then a package for Wendy.
Wonderful.
Yeah.
Okay. Great.
There are some compounds that Wendy could order from a local supplier that were not covered in. There were a couple, because there was one company, Firmenrick, who did not want to send us anything, so we'd have to buy them through a third-party warehouse. But there's one close to Wendy, about an hour or two away.
The Hexarai.
Yeah.
Yes, I know. I will do that as soon as we get a green light when the others will come, so that we can screen them all in one batch.
Uh-huh.
Okay.
And
All right.
probably in a hood, because it will have a lot of smells.
Some of them are quite nice. If I
Yeah, surely the smells are going to be nice, right?
Yeah, but it's just how you mix them. So few nice smells jointly can be awful.
Did what did the Sepios stuff smell like?
That one was okay, but the tea tree oil, when you mixed it with DMSO, you really got high.
Wow. Okay. I'm sure all these features will improve compliance in mass administration programs. From
Yeah.
All right, wonderful. Thanks for that. AOB was to me and Wendy and Ben are coordinating submission to UNESCO call by July 15th. Haven't done anything about that, and we've still got time, so I'm sure we'll get that done. But please leave that as an action item because we need to tell UNESCO about what we've been doing in MycetOS because it's a great example of what they want. And then there was a last good thing about Ian, who came on board last time and was talking about an E. coli-based screening platform for mycetoma targets. It'd be useful to have that conversation. It's not urgent because I think there's an issue there to do with the difference between E. coli and mycetoma, which may prevent that from being useful. But it would be interesting to think about.
This is quite a lot of difference between.
Yeah, so it's not the same thing as a TB screening platform, which is using E. coli, which is the original intention of that. All right. Any other business?
Just a quick update from UCL, maybe.
Yeah, sure.
Just share the screen. Just like to reflect on the few of the compounds that I've been making lately. So the last of the phenarmols that I did a while ago was the one with the quinoline substrate, and DM61 is the last phenarmol that will be sent like in the next batch. So this concludes the whole phenarmol series, at least like for now. For the ketoxymes now is the main series that we are working on, and on this slide you can see the latest compounds that we have been made or are currently in development. Actually, there is a mistake. This should be DM64, sorry, the first one, because I changed this one as well. Yeah. So I have made at the moment, I would say 5 compounds, and 2 are still pending. So either purification stage or I'm making a substrate. But I would expect them to be sent out before the deadline, the 1st of July, which Wendy have outlined as the deadline for the in vivo screening. So hopefully at the end of June, the latest, we will send another batch. I had a bit of issues with low molecular weight substrates, such as like, for example, 1-chloropropane. So I had to change the synthetic route and had to use hydroxyphthalimide as an intermediate. So 2 protocols actually have been adopted like in the preparation of them substrate, and another protocol has been used in the synthesis of the catechozyme. This is opposed to the original one which employs bismuth oxide as a catalyst. And so far it looks everything works perfectly well. So standard reaction conditions for the first reaction would take around like 3 days. But I found like microwave synthesis method which does the job like in under 16 minutes and gives like a proper yield. So this should not be a problem like for the future substrates which have like a low molecular weight or which might be volatile. The second protocol, I'm just about to run it like today, so I'll see how it goes, but it should be okay as well. And it's also usually like around like 2 days, but like this one can be made like in under 5 hours, which is like relatively quick. So the first example would be 1-chloropropane, which has been already done. The yield is 62%, and the next reaction should be done like today, and I'll report on this later on. Apart from that, like 2 more substrates have been proposed. One of the is the azokane, and the other one is the morpholine. And this has been like discussed on GitHub previously, so I'm waiting for those reagents to come in. No, actually, I'm waiting for the chloro reagents to come in and to make the amine substrates, hydroxyl amine substrates for the catechozyme synthesis. We might be able to get this through with the next batch as well, hopefully, but it depends on the delivery times and how the synthesis goes. And lastly, just to follow up like with the catechozymes and the future scaffolds that we talked before, I just would like to say that like I found like a method which is like the fluconazole synthesis as a model synthetic route for the production of new scaffolds. So it would include like Friedel-Crafts acylation, SN2 type reaction in the next step, depending on how we would like to diverge in terms of synthesis and how complex the scaffold would be as the final molecule. We can stop like halfway like through this synthetic route, or carry on like with trimethyl sulfoxonium iodides to make the epoxide ring, and then add another acyl ring if necessary. But this is like still subject to clarification with the SAR campaigns investigation that I'm currently doing for GitHub. And probably that's it for today. Yeah.
All right, great. Thank you.
Yeah.
Mar and Dimitri, the slides you presented, if you could just dump them on the issue for today's meeting, that would be great. Just so we have access to those. All right. Anybody else?
Yeah, Matt,
sorry, just to confirm. Sorry. Oh, sorry, and go ahead.
Just obviously from our side, making our contribution as effective as possible, because we can obviously do the synthesis, but that's quite limited in terms of the students' chemistry knowledge, and we're quite reliant on you guys feeding into us as to really getting us through the synthesis and what's good things to do. So what sorts of things can we do away maybe from the synthesis to help the project and to keep it moving forward? So a big part of my interest is like the awareness and kind of the wider raising awareness of open source research and of the need for this sort of research, and how to promote it in a really good way. That obviously there's some talk about funding and how we can maybe help raise awareness around funding and what's those sorts of things as well. And there's lots of other schools. We've kind of presented this idea at some sort of schools conferences already. There's a lot of other schools that are interested in really getting involved. So how to make that as effective as possible to kind of really help build this project for you.
Yeah, I think if you're interested in sort of like the raising awareness and the sort of like the communication of this project, I think the best person will be actually to speak to Kim, because Kim and some other students from our research group are also quite active in this. I think one of our PhD students Clem is going to sort of like take over this some of these communication aspects as well as part of her research. So I think maybe talk to Kim and some of the other people from the scope group, which is our group here. We should be able to maybe work together or help you with that as well.
I mean, absolutely, yeah, I would agree with that. The most valuable thing remains molecules, and because those are the things that are difficult to get a hold of, and those things that make a big difference. The other stuff though really is also to do with the writing online. I mean, the wiki is always out of date, basically.
Yeah.
And if students can get involved with a mentor like Kim or somebody else,
yeah,
who and they can guide them in what needs to be written up on the wiki, that
yeah,
to make things more up to date, that would really help.
Okay.
Obviously, if the students also want to write up a page about what they've been doing in a way that's easily shareable by us and others, that really helps everybody because it shows what people have been doing. Yeah, those things are very, very valuable.
Bits that they've made, some like infographics and stuff, which Chris has got kind of a wider chemistry heads of departments meeting coming up soon, which I think he might have spoke to you about already. And he's going to share those sorts of things. Yeah, it's more centralizing all as well. But definitely the online writing, they're really keen to do stuff. They're super keen. So the more we can throw at them, it's just kind of the right people to get the information from, to channel it to them, if that makes sense.
Right.
So I'll reach out to Kim.
Yes, cool. Definitely. I think that the scope group. So Kim, I can also partially help. So although I'm not mainly on it, Clem, Alice, we should all be able to either help you or point you to the right direction, because we're doing a lot of these kind of works at UCIT or in my group. So we can definitely help you with that.
Cool. Yeah, that's great. That's awesome.
All right, we're on the hour. Wendy's got another meeting. So yeah, we should probably finish. So it's great to see everybody. Thanks very much for coming along, giving your time. Next meeting, just for the diary, even though hopefully it's already in the diary, is 28th of June, 9 p.m. for UK time. So it's the evening slot for us this time.
Bye, everyone.
Okay.
Bye.
See you again.
Bye.
Bye.
Bye.